Hello everybody. The topic for today is biosimilars. Since biosimilars has garnered a lot of attention these days, I thought maybe we could learn some things about biosimilars. So in this presentation, we'll be knowing what biosimilars are and why do we need biosimilars. We'll also just be knowing few steps that are involved in the development of biosimilars. We'll not be going into the details of it because it's more of a preclinical thing. Then we will be seeing various biologics and biosimilars in the clinical practice. We'll also be seeing what interchangeable biosimilars are, and we will see the differences between biosimilars and generic drugs. So, what are biologics? They refer broadly to substances produced from living cells. So, using biotechnology, and there are various biotechnological methods like recombinant DNA technology, production of vaccines, proteins, hormones, etc. Whereas for the production of monoclonal antibodies, you have hybridoma technique. Okay, using various recombinant DNA technologies or antibody technology, you develop something out of a living organism. It is biologic, and you can see the various examples like hormones, proteins, cytokines, okay, cellular and gene therapies. All of them will come under biologics. Now, what does biosimilar mean? Biosimilars are also known as similar biologics, and these are the medicines that are similar to other biologics which has already been authorized for use. So there is a biologic that is already being used, and you develop a similar molecule for the biologic. It is called biosimilar. Okay? And to be even more specific, the New Drugs and Clinical Trial 2019 it defines biosimilar as a similar biologic because it is called a similar biologic also. Biosimilar is otherwise called a similar biologic. And what does biosimilar or similar biologic means? It's a biological product which is similar in terms of quality, safety, and efficacy to reference product licensed or approved in India or in any other approved ICH countries. ICH member countries, as in there are few other countries that are part of ICH. ICH means International Conference on Harmonization. And these biosimilars are known by various names. They are called as biosimilars in Europe, and they're also called as follow-on pharmaceuticals in the United States and Japan, and they're called as subsequent entry biologics in Canada, biocomparables in Mexico. In India, biosimilars are referred to as similar biologics. So the first biosimilar that is developed in India is a biosimilar of hepatitis B vaccine. Whereas in the United States, it is the biosimilar of filgrastim. Filgrastim is a colony stimulating factor which can be used in the treatment of neutropenia. And in Europe, the first biosimilar that is developed is of recombinant growth hormone, and it is called as Omnitro. Now, what makes biosimilars unique? Why is this biosimilars of much interest these days? Is if you see, these biologics generally exhibit higher molecular complexity. You can see erythropoietin here and aspirin. Erythropoietin is way more complex than aspirin is. The aspirin structure is simpler when compared to this complex structure of erythropoietin biologic. And not only that, not just the complexity, they are produced from cells in culture or they are also produced from whole organisms. So even minor change in the process can cause a very major change in the product. Unlike the production of normal pharmaceuticals, where minor change in the process might not be able to cause major change in the product. It might not exactly translate to the major change in the product. Whereas that is not the case for biologics. In the process, even a smaller mistake could end up getting a major outcome. It could end up getting a major change. So that is why, because of their molecular complexity and because of the strenuous process that is involved in their development is unique. So what biosims we are referring to them as biosimilars. So how similar should they be? So they'll define the similarity as no clinically meaningful difference. So there should be no clinically meaningful difference from the reference product in terms of potency, safety, efficacy, and immunogenicity. Now, why do we need biosimilars? The main reason for the development of biosimilars or for the need for biosimilars is cost reduction. We all know biologics are costlier when compared to pharmaceuticals. Okay, because of their complex structure, because of the complex process that is involved in their development, they are expensive. Since they are expensive, we must develop biosimilars to the biologics, just like how we develop generic drugs to the original branded drugs at a much lower cost, so that these biosimilars will be available to patients who cannot afford maybe biologics. Not just cost reduction, we all know every drug has patency, biologics has patency. Once the patency expires, there must be another way in which that particular drug or biologic must be available for the patient. So only if we develop biosimilars, we will be able to cater to the needs of the patients even after the expiry of the patency. So once you end up developing biosimilars, either for cost reduction purposes or after the patency expires, you are essentially creating a competition in innovation wherein companies will have to work towards producing biosimilars in a most cost effective way. And you do not have to worry about the safety and efficacy just because of their cost effective nature. Even though they are cost effective, the regulatory authorities, be it in the West or in Asia, they are very stringent uh, in terms of approving biosimilars. So generally they'll be maintaining safety and efficacy to the reference biologic. So on that aspect, there need not be any worry. And if you see, this is showing how cost effective can development of biosimilar be. If you see, for development of new biologic, it costs around 1.2 to 2.5 billion US dollars. Whereas for biosimilars, it is around 100 to 200 million, which is substantially low when compared to developing a new biologic altogether. Just by encouraging or involving in the development of biosimilars, United States is able to save $18 billion from 2010 to 2014 and $19 billion from 2015 to 2019 and $104 billion from 2020 to 24. This is clearly telling the improvement or the increase in the rate of manufacturing of biosimilars for the past 3 to 4 years. So I'm going to just skip these steps because these are not of much clinical importance. It's of rather how they are developed. So for developing biosimilar, you will first need to select the reference product. For example, if you want to develop a biosimilar to insulin glargine, the insulin glargine would be your reference product. And once you select the reference product, you will physically and chemically and functionally, in all possible ways, you will characterize it. You will characterize its molecular structure, its size, its charge. You will know its biological activity. You will see if it has any post-translational modification or if it is able to undergo post-translational modifications like sugar molecules on the protein, etc. Because this is also important. You will see all of this because only when you know, when you have the data of all these about your reference product, you will be able to develop a similar product to it, right? So that's why you will analytically characterize it. Once you analytically and this is how you characterize them. These are various techniques, be it mass spectrometry, be it X-ray crystallography, be it sample tube, or be it plasma surface plasma resonance. With all this, you will characterize it. These are various techniques used to characterize it. You do not have to know in detail about it. Once you have characterized it, you will develop a cell line. Remember what I told about biologics? They are generally produced from living organisms or cells. So you should develop a cell line that is capable of producing your biosimilar. So for that, you will have to choose an appropriate host cell. Generally, they choose CHO cells for monoclonal antibodies. Whereas if you want to develop vaccines, there is human embryonic kidney cells. Uh, Actually, regulatory authorities also have guidelines Depending on what type of biosimilar you will develop, they have guidelines on which cell lines you should be using for the development of your biosimilars. And factors to be considered is, like I said, post-translational modification is really important. If your cell line is capable of producing molecules that can undergo post-translational modification, because post-translational modification does play a role in the efficacy of your biosimilar. So that is important. You should choose a cell line that is able to produce biologics which can undergo post-translational modification. And you should also keep in mind the yield that your cell lines are producing, because once you are producing biosimilars, you are doing it in a large scale. So your cell lines must be capable of producing it in a large scale at an industry level, not just at a bioreactor level in your laboratory, but at an industry level, they must be able to produce large yield of your biosimilars. And like I've already said, because there are regulatory guidelines on how you choose cell line for the development of biologics, you will have to adhere to them also. And this is a Chinese hamster, and these are the Chinese hamster ovary cells, and this is human embryonic kidney cell. And this is various steps that are involved in the biosimilar manufacturing. Not going into detail of it. Industrial bioreactor where this biosimilars are produced. Again, this is steps comparing above one is the reference product and below one is the biosimilar. And once you produce it with all the steps, you will obviously carry out preclinical studies to determine its biological activity, to determine the pharmacokinetics and dynamics, and to determine the toxicity and immunogenicity. You can conduct both in vitro and in vivo studies. And of course, you will do clinical trials. In clinical trials, the thing you should remember is you will generally not do phase two trials, because the point of developing biosimilar is to reduce the cost. To reduce the cost, if you can cut down at least one phase, it will be a value of it will be much cost effective. So you will not do phase two, you will do phase one, and then directly you go for phase three. You will just confirm it. You will not do phase two, and of course phase four there is pharmacovigilance will anyway be there. So once you have the data of both your analytical, preclinical, and clinical, you will submit it to the regulatory authorities, and they will grant you permission. That is how you developed. And once you developed, these are now we have these examples of biologics and biosimilars that are in clinical practice as of today. Of course, there are many other, but these are few. I felt these are important for us to know. So the first one being the biosimilars for bevacizumab. We all know bevacizumab is an antivascular endothelial growth factor. So it is used in the treatment of cancer, where it can act against angiogenesis. Angiogenesis is a essential part of the pathology of carcinoma, right, for the development of carcinoma. So to inhibit the angiogenesis, we use bevacizumab in almost metastatic colorectal cancer or non-small cell lung cancer, metastatic renal cell cancer. Okay, and the biosimilar being of bevacizumab is BVZR, bevacizumab BVZR, bevacizumab AWWB. If you see the last four-letter suffix, that is characteristic last four-letter suffix that is given to the biosimilar just to identify it as biosimilar. So if the drug has last four-letter suffix, that means it is biosimilar of the reference product, your bevacizumab. If it does not have any four-letter suffix, that means it is the reference product. Just with this code, they will generally recognize. But however, these are all what to say, non-proprietary names. You will have brand names anyway, proprietary names anyway. So clinicians usually remember them through brand names. Likewise, you have different types of biologics and biosimilars that are being developed. You can have a look at all of them and the conditions where they are used. Next list of pegfilgrastim, like I've said, these are colony stimulating factors and are used in the treatment of neutropenia. Rituximab, anyway, no, and biosimilar that is developed is insulin glargine. For insulin glargine, there is a biosimilar of insulin glargine IFGN. It is a long-acting human insulin. is for diabetes mellitus. So all these are important actually. Infliximab, rituximab, insulin, pegfilgrastim. In the previous one also, if you see, okay, so in the previous one also there are a few important things you can go through them. And this is just another way of characterizing these all these biosimilars in the respective branches that they used. What are the biologics and biosimilars that are used in immunology, oncology, supportive care, like filgrastim, eptoin, okay, and insulin in ophthalmology. Okay, this is just another way of characterizing them. These are some of the biosimilars approved in India. So, like we have seen how they are used, how they are used, how are they produced, and why do we need biosimilars? Just like any other drug, they also have some challenges or adverse effects. The main being immunogenicity. Since these are biologics, since these are obtained from living cells and they're given to the patients, it's natural that there will be immunogenicity. But that turned out to be the feared complication. Here in this case, if you see, is causing pure red cell aplasia associated with a biosimilar of eptoin alpha. Generally, eptoin alpha we give nothing but recombinant erythropoietin. We generally give it for the treatment of anemia. If itself can cause pure red cell aplasia, so what is the point of treating the patient with eptoin when the patient has already anemia and you're giving it to treat anemia? So such kind of immunogenicity is not acceptable. Even even otherwise, immune reaction also might not be acceptable. But affecting the purpose of treatment altogether is definitely not acceptable. So immunogenicity has been a major concern for biosimilars. Another example being human albumin stabilizer also. Uh, like recombinant, so human albumin stabilizer is also basically they're talking about eptoin alpha only. In eptoin alpha, the albumin stabilizer was replaced by polysorbate 80 and glycine. So initially there was one human albumin stabilizer. They have replaced it with polysorbate 80 and glycine. So that led to the pure red cell aplasia. Like I said, even just like how excipients are important in generic formulations, here also the stabilizers or the other things that are added in addition to the active ingredient is important here as well. Okay. So in another study, they say the presence of neutralizing antibodies were found when patients were treated with recombinant interferons. And these antibodies ended up destroying interferons. Again, uh, same example as instead of producing interferons, the patients, instead of helping in the function, these interferons produced autoantibodies that ended up destroying the present interferons in the body. So, immunogenicity in this way is very concerning. Just like any other drug, it also needs pharmacovigilance, especially all the adverse effects, or just like how all the adverse effects of drugs are being reported for biosimilars also, they must be reported. And what is interchangeable biosimilar? Interchangeable biosimilar means, in addition to all the before required data I have told, these must be showing evidence in switching studies. That is, you give the patient original biologic, and you after a while you can switch the patient to biosimilars. And even when you switch the patient to biosimilars, those biosimilars should be producing similar effect just as the original biologic was producing. So if you can able to demonstrate that data to the regulatory authorities, they will give a interchangeable status to the biosimilar. So once the biosimilar gets the interchangeable status, the pharmacist, generally it is not practiced in India, but in Western countries, the pharmacist will be able to switch. He'll be able to give biosimilar even if the prescriber writes biologics, and that means he'll be free to give biosimilars if there is no stock of biologics. He can give biosimilars. So that is what interchangeable biosimilar means. That is what interchangeable status means to a biosimilar. So this is another last slide showing the difference between biosimilars, biologics, and generic drugs. If you see, I'm not going into few details. The main difference you must remember, both biosimilars and biologics, they undergo stringent regulatory process. The cost is also more. For biosimilars, it might be little lesser than biologics, but it is still more. Um, there will be stringent regulatory requirements for its development. Whereas for generics, it's not same. The expense is also less. Generally, branded drugs are little relatively less expensive than biologics. So generics are even more less expensive, and there will be no stringent regulatory authorities also, or just because there is no stringent regulatory authorities, and the process also for development of generics is very less difficult when compared to the development of biosimilars. With all these reasons, generics are relatively less expensive. So that is the main difference you have to take from this table. So these are my references.